Hello and welcome to Nikolay's genetics lessons, and today we are going to talk about laboratory calculations. We are going to talk about solutions, dilutions, and dilution factor. So here's a problem. 2 milliliters of serum sample was added to 18 milliliters of the phosphate buffered saline PBS in tube 1. 10 milliliters of tube 1 was added to 40 milliliters of PBS in the tube 2. What is the dilution of the serum in the tube 2? So here's the tube number 1. Tube number 1. In this first test tube, we put 2 milliliters of the serum sample and added 18 milliliters of the PBS. So 2 milliliters plus 18 milliliters. And we are going to get 20 milliliters. And concentration is going to be 2 divided by 20, or 1/10. So dilution factor is going to be 10, and dilution is going to be 10-fold. As for the second tube, so tube number 2, we took 10 milliliters from the tube number 1, so 10 milliliters, plus we added 40 milliliters of the PBS, so plus 40 milliliters would give us 50 milliliters. And the dilution is going to be 10 initial milliliters divided by the total amount, which is 50 milliliters. So we are going to get 1/5 here. So we diluted here 5 folds. Now we have just to multiply these dilutions, which are serial dilutions. So 1/10 is dilution in the first test tube, and we have to multiply by dilution 1/5 in the second test tube, and we are going to get 1/50. So dilution is going to be 50-fold. Or we also can say that as a result of these serial dilutions, we diluted 2 milliliters of the serum sample 50 times. And this is all for today. Subscribe and see you in the next video. Goodbye.